Hello students, welcome to Karnataka State DCLMS e-learning platform. In today's session, let us learn about microscopy. In the previous session, we have learned about microscopy, microscope, the principle behind microscopy with special reference to magnification and resolution. In today's session, let us learn about the different types of microscopes. Types of microscopes. Microscopes are designated as either light microscopes or electron microscopes. Light microscopes uses visible light or UV light to illuminate the specimens, and examples include bright field microscope, dark field microscope, phase contrast microscope and fluorescent microscopes. Whereas electron microscopes uses electron beams instead of light rays and magnets instead of lenses to observe submicroscopic particles. Fluorescent microscope, though is a light microscope, uses UV light instead of light to illuminate, and the advantage is that UV light has a much shorter wavelength when compared to that of visible light and is not directly perceptible to the human eye. Let us learn about light microscope in detail. A light microscope is a laboratory instrument or a tool that uses visible light to detect and magnify very small objects and by enlarging them. They use lenses to focus light on the specimen, magnifying it, thus producing an image. The specimen is normally placed close to the microscopic lens. Microscopic magnification varies greatly depending on the types and number of lenses that make up the microscope. The lenses are aligned in that they can be able to bend light for efficient magnification of the image. Now, depending on the number of lenses, there are two types of microscope, that is simple microscope and compound microscope. In simple microscope, it uses only single lens and therefore it has low magnification. Whereas compound microscope has higher magnification when compared to simple microscope because it uses two sets of lens, an objective lens and an ocular lens, or also known as eyepiece. The functioning of the light microscope is based on its ability to focus a beam of light through a specimen which is very small and transparent to produce an image. The image is then passed through one or two lenses for magnification for viewing purpose. The transparency of the specimen allows easy and quick penetration of the light. If at all the sample is not transparent, then the light cannot pass through the specimen. Therefore, it doesn't become available for the lenses to magnify. So therefore, transparency is very important. So whatever is used in microscope, we ensure transparency. Be it slide, a clean glass slide which is transparent in nature, a clean cover slip which is again transparent in nature, and a thin section of the specimen or thin smear of the specimen or culture ensures that the light passes through unhindered. Therefore, it becomes easier for viewing. Next, specimens can vary from microbes to cell to even tissues. Now, moving on to the types of light microscopes. The modern types of light microscopes include bright field light microscope, phase contrast microscope, dark field microscope and fluorescent light microscope. Since in the syllabus it is mentioned that we have to learn only about phase contrast and fluorescent microscopes, let us focus only about them. First one is phase contrast microscope. So this is a type of optical microscope where small light deviations known as phase shifts occur during light penetration into the unstained specimen. So the specimen used here is not stained, it is unstained. Now, these phase shifts are converted into image to mean. When light passes through the opaque specimen, the phase shifts brighten and the specimen forming an illuminated image in the background. That is, if you can see the example which I have given here, that is the contrast between oxytricha. See, both are the same oxytricha, but the first one is observed under normal compound light microscope and the second one is observed under a phase contrast microscope. So you can make the difference. So there is a starking contrast between the background and that of the organism. So it seems that the organism is highlighted because of the phase contrast or because of the phase shift. So that's the advantage of using phase contrast microscope. So this PCM or phase contrast microscope produces high contrast images when using a transparent specimen, more so of microbial cultures, thin tissue fragments, cell tissues and subcellular particles. So this is how phase contrast microscope looks like. Everything is same except for a filter which provides the phase shifts. So otherwise it mostly resembles that of any compound LED microscope. The principle behind the working of phase contrast microscope is the use of optical method to transform a specimen into an amplitude image that's viewed by the eyepiece of the microscope. The phase contrast microscope can be used to view unstained cells, also known as the phase objects, which means that the morphology of the cell is maintained and the cell can be observed in their natural state in high contrast and with efficient clarity. So this is because if the specimens are stained and fixed, they kill most cells, a characteristic that is uniquely undone by the bright field light microscope. In case of bright field, what happens is the specimens are being mostly killed in order to view, whereas in case of phase contrast it can be left live. So one need not stain, because the very moment it is being stained, the cell would not stay in its leaving state. So therefore in case of phase contrast it can be observed live. The shifts occur during light penetration, become converted to changes in amplitude, which causes the image contrast. Coupled with contrast enhancing elements such as fluorescence, they produce better visuals of the specimen's image. So in the image you can see the path of light flowing and the image formation. The only difference is that it has a face plate which allows the formation of phase shift. Next type is fluorescent microscope. So in the case of fluorescent microscope, the specimen emits light by adding a dye molecule to the specimen. So this dye molecule will normally become excited when it absorbs light energy. That is, in this case it is the UV light, wherein upon absorption by the dye molecule gets excited. So hence it releases any trapped energy as light. The light energy that is released by the excited molecule has a longer wavelength when compared to its radiating light. The dye molecule is normally of fluorochrome that fluoresces when exposed to light of a certain specific wavelength. The image formed is a fluorochrome labeled image from the emitted light. So in the image you can see how a fluorescent light microscope looks. So the only difference here is that instead of visible light we are using UV light, and upon UV light emission the fluorochrome molecules, they get excited. So whatever energy is there within them will be released. The trapped energy will be released as light. So that is reflected and that is what is seen. So it can also be coupled to a camera and be viewed on the monitor. Next, the principle behind this working mechanism is that fluorescent microscope will expose the specimen to ultraviolet or blue light, which forms an image of the specimen that is emanated by the fluorescent light. They have a mercury vapor lamp that produces an intense beam of light that passes through an excitation filter. So the excitation filter functions to transmit a specific wavelength to the fluorochrome stained specimen, producing the fluorochrome labeled image at the objective. After the objective, there is a barrier filter that functions primarily to remove any UV radiation that may be harmful to the viewer's light, thus reducing the contrast of the image. So basically the wavelength of light involved is UV, but since UV can damage the human eye, after the image is formed, it has been filtered. The UV light is filtered so that it does not affect the viewer. Moving on to electron microscope. Electron microscopes, they differ from light microscopes because they produce an image of the specimen by using a beam of electrons rather than beam of light rays. So electrons have a much shorter wavelength than that of visible light, and this allows electron microscopes to produce higher resolution images than the standard light microscopes. Electron microscopes can be used to examine not just whole cells but also subcellular structures and compartments within them and produce very high resolution images. So the limitation when it comes to electron microscopes is that the samples must be placed under vacuum in electron microscopy, and it is prepared via extensive fixation process. So live cells cannot be imaged with the help of electron microscope, so they have to be killed for viewing. So that is the disadvantage or the limitation of electron microscope. So here in the picture you can see Salmonella bacteria slide smear. In case of a light microscope, whatever is seen in the left is how it is seen under light microscope, but whereas the image on the right, we can see the highly magnified image of a single bacteria. So that is the level of magnification that can be achieved through electron microscope. Here they have used scanning electron micrograph, and the best part of this image is that we can even observe that the bacterial cells are being attacked by the body cells. So that can be seen because you can see the bacteria being strangled. So even to that extent it can visualize. So that's the advantage of electron micrograph or electron microscope. Now there are two major types of electron microscopy known as SEM and TEM. SEM stands for scanning electron microscopy and TEM for transmission emission microscopy. So this is the image of electron microscope. So electron microscope is not very handy and it cannot be placed on the desk. It's a very huge one. It almost resembles the size of an industrial gas stove, and it consumes a lot of space. So it is very bulky. Next, in scanning electron microscopy, that is SEM, a beam of electrons moves back and forth across the surface of the cell. As the name suggests, scanning electron microscope, it only scans through the surface. So this creates a detailed image or 3D image of the surface. So this type of microscopy was used to take image of Salmonella bacteria that is shown before. So here in this slide you have seen the Salmonella bacteria. So it is obtained using scanning electron microscopy. Next, in transmission electron microscopy, in contrast, the sample is cut into extremely thin slices using a diamond cutting edge, say for example, before imaging. So the electron beam passes through the slice rather than skimming over its surface. So it is not just going to skim over the surface, rather it's going to pass through the specimen. So TEM is often used to obtain detailed images of the internal structures of the cell. So SEM mostly gives the surface view, whereas TEM gives even the internal structure details also. So electron microscopes are significantly bulkier, they're very expensive than the standard light microscopes, but they can be used to study at subatomic level. So there lies the advantage. To quickly summarize, light microscope is an instrument for visualizing fine detail of an object. It does this by creating a magnified image through the use of a series of glass lenses which first focus a beam of light onto or through an object, and convex objective lenses to enlarge an image formed. Phase contrast is used to enhance the contrast of light microscopy images of transparent and colorless specimens. It enables visualization of cells and cell components that would be difficult to be seen using ordinary light microscope. Phase contrast does not require cells to be killed, fixed or stained. Whereas fluorescent microscopy is an imaging technique used in light microscope that allows excitation of fluorophores and subsequent detection of the fluorescent signal. It is widely used due to its high sensitivity, specificity and simplicity, and it can be applied to living cells and organisms. In scanning electron microscopy, a beam of electron moves across the surface of a cell, whereas in case of transmission electron microscopy, the electron beam passes through the slice rather than skimming over its surface. Thank you for watching.